Hi everyone! For Chapter 12, we are going to talk about how gene expression is controlled. To go from genotype to phenotype, we have gone over the process of transcription to make mRNA molecules that carries the instructions on how to make a protein. And we have gone over the process of translation, which is the actual construction of the protein by ribosomes. But it is very inefficient for our cells to be chunking out gazillion different types of proteins when we don't need all of them at once or at the same time. Just because we have hundreds of recipes of all the cakes we can bake, we won't go and make hundreds of cakes at the same time. Remember that my analogy of going from your DNA recipe to your final protein structure which is analogous to kind of baking a cake from a bunch of recipes. So it doesn't make sense for us to bake all hundreds of those kind of cakes at the same time. It is a waste of energy and resources. So same thing for our cells. Hence, scientists know there has to be some way for our cells to regulate when and how much of each protein we need in order to keep us alive, yet conserve energy. This is called gene regulation, which is what we're going over in this chapter 12. It's a relatively short chapter because we're only going to talk about eukaryotic gene regulation. Now, there arise two situations already that we can think of that we will require some sort of gene regulation if we think about it. One of those we have already talked about. Remember, we have discussed genes that are expressed under different environments. The rabbit with the dark pigmented ears and feet when they're reared at lower temperatures. We call that the temperature-sensitive allele when we were talking about all those different genetic inheritance patterns. That particular allele was only active at a certain temperature, which means it is not expressed 100% of the time. How do we go? How do we go about controlling that? That is the question. Epigenetics is another of those topics we talked about related to gene regulation, that how our genes is expressed is highly relevant to the environment of the organism sometimes. Another even more regulated or related situation is for us as a multicellular organisms. All of our cells has the same set of genetic information, but yet not every cell perform the same task or make the same type of proteins. For example, our neurons are cells that make neurotransmitters for transmitting messages between neurons so we can have reflexes and voluntary movements. But our white blood cells don't make neurotransmitters. They make antibodies and toxins that help kill pathogens. So our muscle fibers have actin and myosin for contraction, and we know our bone cells don't make those proteins. Yet all of these cells that I just mentioned contain all the genetic and same genetic material within that same organism or individual. So there has to be some mechanism that exists, especially during early embryo development, for different cells to become specialized for their specific jobs. How is this achieved? First of all, our genes can actually be divided into two categories. One is called the structural genes. These genes are used to encode proteins that are actually used for that specific function, whether it be metabolism, biosynthesis, or some sort of structural role in a cell that we've mentioned, all that the proteins do, okay? And as we have mentioned about genetics early on, things are always more complicated the deeper we dive into each topic. Not every gene is regulated in the same fashion. Different genes will be controlled differently. How often they're turned on is different. How much protein products each gene makes is different. And at which rate or at what rate is the protein being made is different. So There's always genes that are always turned on as well, okay? Because not every single gene in your body must be timed. There are what's called essential genes that perform very important everyday, every minute cellular respirations or cellular functions that we have to turn those genes on all the time. Those are called constitutive genes, okay? Constitutive genes for those that have to be turned on all the time. So, for example, ribosomes are the perfect candidate to be a constitutive gene because they are proteins themselves. But yet, without them, we can't do any protein synthesis at all. We're relying on ribosomes to be chunking up proteins for us every single minute. And hence, those are the kind of genes we're going to keep reading all the time, and we don't need for it to turn off at any given amount of time, or else everything just stops for protein synthesis. So those are one of those proteins that are constantly being made in our cells, okay? Ribosomes. The second category of genes are called regulatory genes. Hence its name regulatory, because these are genes not in charge of encoding a protein. They will perform enzymatic they are not the ones that's going to be performing those enzymatic reactions for you or contribute in any structural roles. They're gene products, okay? Gene products, which for this case can be either RNA or proteins. They are the ones that will interact with other DNA sequences or other genes and play a role in controlling those genes' transcription or translation rates, or pretty much means they can turn on a gene or turn off a gene. So there are portions of our DNA sequence that have these so-called regulatory genes that control how our structural genes are being made, okay? Or expressed. So if you go to this diagram here, okay, it shows you an example where here, if you have a regulatory gene somewhere in your DNA sequence, regulatory gene do not encode for a protein that functions as an enzyme or whatever it is, okay? It is just purely for control of your structural genes. So that's why after transcription, you may end up with some sort of regulatory protein, and this regulatory protein can then act on a structural gene, either turning it on or turning it off, okay? We'll talk about a little bit of it, but we won't go into detail as to how those operators work within your bacterial cells, etc. Okay, if we get into those really detailed stuff, it's just too much to mention, and hence we're just going to stick on the basics. Introduce you what it is that regulate gene expression. Okay. So sometimes this regulatory protein will turn on RNA polymerase or allow RNA polymerase to bind, which then allows your gene to be transcribed, whether it be gene A, B, or C. And then with these structural genes, then you're going to make mRNA from these genes, which then can become their, potential protein A, B, and C, okay? And so the biochemical pathway is we go from some precursor to some intermediate product and a final product in the end, okay, when we talk about this process of going from a gene to a protein, okay? Now, in addition to regulatory genes, there's something called regulatory elements, okay? These sequences in the DNA are not even transcribed at all, okay? So remember, regulatory genes are the ones that actually get transcribed into a regulatory protein or stay as an RNA molecule that's going to affect your structural genes. But structural elements, we're not even talking about any transcription at all, okay? They are just sequences, okay, that provide a binding site for other regulatory proteins to bind, so it either activates transcription or block transcriptions of a gene, depending what that regulatory element is for. We'll talk about examples of these as enhancers and insulators when we get to that part of the chapter. So now that we understand chromatin structure all the way down to how translation is done, we can discuss at what points can gene expression be regulated. Okay, there are 6 different levels where we can alter gene expression, and it goes in the order of chromatin structure, transcription, mRNA processing events, and translation. And we're going to talk about all of those within this diagram here in this slide and the next slide as an introduction. Some of them I will dive a lot deeper into because we haven't mentioned it before, and there are some that we already talked about, so just quick gloss over what they are, or reminder of what they are again. Okay, so first we can control gene expression by changing the chromatin structure of DNA. This includes modifications made to histone proteins or modifications that's been made to DNA nucleotides themselves. I'll make that distinguish, or what's the difference between those two, when we get to talk about the chromatin structure in detail. We will spend a lot of time talking about different types of chromatin structural conformation alteration because we haven't really mentioned that, okay? Some of the examples we'll talk about is DNA methylation and chromatin remodeling, okay? Changing how those DNA is wrapped up around histones, okay? Second, we can control gene expression at the transcription level. Remember that we have mentioned the assembly of transcription apparatus on promoters that are necessary for initiation of transcription. Part of that transcription apparatus includes a group of proteins called transcription factors. There are transcription activators and repressors that can actually turn on or off a gene. Again, I'll come back to this idea when we get to that part in the lecture, okay? Because we kind of glossed over transcription factors and what makes up that initiation apparatus during transcription when we talked about transcription in chapter 10, okay? All right. Third, it is a level of control that we have already talked about in previous chapters, which was the level of mRNA processing. So this one I'm not going to go too deep into again this time, just a quick review when we get to that part in the lecture. But remember, we had to add a 5' cap and a 3' poly A tail on the mRNA, because from what we have after transcription in eukaryotic cells, they are just known as pre-mRNA. In order for them to become mature mRNA molecule, you had to add a 5' cap and you had to add that 3' poly A tail. And both of those, we already mentioned, play a big role in stability of the mRNA in the cytoplasm or transport of the mRNA molecule into the cytoplasm. Now remember that, during mRNA processing, it also involves removal of introns. Remember, there were introns versus exons. Now, introns have to be removed in order for the pre-mRNA molecule to become a mature mRNA. And there was this idea of alternative splicing that we have introduced in that chapter to talk about how sometimes we don't need 10 different genes to encode for 10 different proteins. Just from the different combination we can have with introns and different exons recombination, you can have different protein versions from one mRNA molecule. So we have already mentioned that from chapter 10. Also, again, I'll gloss over it when we get there, okay? So fourth, we can directly regulate mRNA stability by using RNA interference molecules or RNAi molecules that can lead to either degradation of mRNA that have been made, or less of the mRNA is available, or even control transcription. Okay, either case, less mRNA, less transcription. The result is you will have less amount of proteins that are made, which then controls how much gene or how much of that protein is made from the gene. Okay, so we already briefly talked about microRNA and siRNA. Now this chapter we're going to dive a little deeper into how siRNA and microRNA functions. Fifth, we can control gene expression at the level of translation. Remember that in order for translation to proceed properly, there must be adequate amount of enzymes, protein factors, RNA molecules, and availability of amino acids themselves. So if any of these aren't available, then you can't do any translation. Then lastly, we can control gene expression by post-translational modification. Again, something which we have already mentioned last chapter briefly as a last part of that chapter. Some proteins are only activated when it has been trimmed or cleaved, as I've mentioned before, and some proteins can be activated or deactivated by adding different chemical groups to the amino acids within the protein. For example, methylation and phosphorylation are some of the two common modifications you can do or add to the protein to change its functional properties. Even though you're making the protein, if you're not activating the protein or if you're deactivating the protein, you're not getting a gene expression. Gene expression means the protein has to be functional in order to do its job. Okay? All right. Okay, so let's start with the types of modifications that can happen to DNA's chromatin structure. We talked about how DNA is a double helix in structure, but in order to condense it down so we can fit billions of these base pairs into small little nucleus inside our tiny little cell, DNA is coiled around what's called histone proteins. And when DNA is wrapped around histone proteins, it will not allow transcription to proceed if the promoter or the gene sequence is actually wrapped around the histones, because all those parts of the transcription apparatus we talked about has to bind to those specific sites on the DNA. Okay? RNA polymerase can't access the gene if it's wrapped around the histones. Hence, DNA has to be uncoiled from the histone proteins in order for transcription to start. Okay, so there are things that can make histones wrapped or coiled around DNA more tightly, or there are things we can do to loosen that up so the gene becomes accessible. Remember that when we have, histones like this, and then the DNA wrapped around it, DNA wrapped around it, DNA wrapped around another histone, we see this bead-like structure, like a string with beads under the electron microscope. We call this the nucleosome, if you remember from our DNA structure chapter. And hence, we're going to have to play around with the nucleosome structure when we have to do transcription. Now, there are different ways to alter chromatin structures. Okay, first one is called chromatin remodeling. So there are three. We're just going to discuss the first one, which is involving how we're going to change that nucleosome structure. This is when transcription factors, regulatory proteins, as I've mentioned, those are collectively called chromatin remodeling complexes because they bind to specific sites on the DNA and able to shift positions or reposition of the histones, okay, and somehow they can change the nucleosome overall shape to reveal those promoter regions and genetic regions, okay. When that happens, you can have then your RNA polymerase, your transcription apparatus, all that stuff to come and bind and initiate the transcription process. So this diagram here is illustrating that process. Here you have the histones. So coiled around this histone, for example, is this red region that contains the promoter site. Okay, maybe red and green somewhere that has to do with the initiation of gene transcription. So now, if we have something called the chromatin remodeling complex that can rewrap these histones somehow, turning this one a little bit, turning this one way a little bit, turning this one this way a little bit, then it can actually reposition these histones and nucleosomes so that you can reveal that important section, the red and the important green sections, for your transcription factors to bind. For example, RNA polymerase is one of those important enzymes that must bind in order for it to start making mRNA molecules. So just by repositioning these nucleosomes, we can potentially reveal what's critical for that particular gene to be expressed. Okay? All right, so second way is called histone modification, and there's a handful of ways to modify histone proteins. Histone proteins contain two main domains. One is called the globular protein domain. We didn't really distinguish this when we were talking about DNA structure because it wasn't necessary at the point, but now we need to talk about these two domains because this is where histone modification comes in. So one is the globular protein domain, and the other is a positively charged tail for interaction, okay? Because histone wraps around the DNA molecule. Well, how does it know where to wrap around? How does it know to stay coiled around the histone or the DNA molecule? It's because here the tails of each of these histones kind of like grabbing on to the DNA molecule because they are attracted to it. The end or the tail ends of these histones are positively charged. And we know that DNA molecules are negatively charged. So these tails right here at the end is attracted to the negative charge of DNA, and hence it collapses on the DNA, coiled DNA like this in this figure. Okay? So, because the chemical property of the tail is what's critical for a histone for DNA interaction, it is often what's modified in order to alter gene expression. That means we usually modify the tails of the histone so that it has a different charge, or it blocks the interaction of the tails, the histone tails, with the DNA molecules. Some modification can cause histones to bind even more tightly to DNA, while others can cause histone to detach from the DNA molecule. And we'll talk about examples of these. The types of modification that can happen usually involves adding a chemical group like phosphates, methyl groups, acetyl groups, or even small molecules called ubiquitin. Okay, the process of adding ubiquitin is called ubiquitination. Okay, ubiquitination. So this one is a little different from just adding little chemical groups. This, you're adding small molecules to the histone to change its interaction with the DNA. I won't go deep into that, but just know that there are these different types of modifications that can happen on the histone that will modify how DNA is interacting with those proteins. Okay, so these modifications collectively, we call it histone code because they can affect how genes are expressed. Sometimes these modifications are changing chromatin structure directly. Sometimes it can be indirect effect by providing recognition sites for transcription initiation proteins to bind, for example. So if you see here, here are the tails of the histone protein, okay? Histone protein, and it is not acetylated yet. Once you add an acetyl group to the tails of these histone groups, somehow it caused a relaxation of the coils around the DNA molecule that you see that there's a much bigger gap exposed between the histone proteins compared to when there was no acetyl groups attached to the tails. So this can potentially expose a promoter region or a whole gene for that matter for the transcription to begin, okay? So sometimes these modifications will work differently, okay? So for example, okay, we'll talk about two specific examples. One is where we add methyl groups. The other is when we add acetyl groups. So methylation of histone is when we're adding methyl groups or a CH3 molecule to the tails of the histone. This effect can either be activation or repression of gene expression, depending which histones gets methylated, how many of them gets methylated, and sometimes it differs even by which amino acid on the histone was methylated. So methylation can have a wide variety of effects, okay, depending on those things. But the enzyme that does this modification is called histone methyltransferase. Very easy to remember. It acts on histones histone methyltransferase. Okay, it acts on histone and it transfers a methyl group to the histone. So that's why it's called histone transferase. Okay, it transfers a methyl group to a histone protein. Now, there are enzymes that are capable of removing methyl groups to change gene expression. This was what I was circling earlier erroneously. Okay, so that's the histone demethylases. These enzymes are called histone demethylases. It acts on the histone. It actually removes a methyl group from the histone. Okay? This is an example of a diagram of what may happen if methyl groups were added to the tails of the histone. So imagine these are your histone proteins, all these blue balls, and then the tails coming off of it is that histone tail that has been methylated. Okay, methylation are represented by these green dots here. Okay, and in this particular case, the methylation of histone caused a tighter wrapping around the histone protein, such that we are really embedding and tightly coiling our gene around the histone. So if an RNA polymerase was to try to access that, it can, okay, because it's already wrapped around the histone. So any transcription factors or initiation transcription apparatus, they cannot access that particular gene because it's wrapped around the histones. So another example is where we're adding a acetyl group or CH3 with a carbon and an oxygen. Those are added to histone and we call that acetylation of histones. For acetylation, this usually results in activation of a gene. Okay, so this is different from methylation. Methylation can go either way, activation and repression, depending on how it modifies the histone. For acetylation, however, it usually results in activation of a gene. Rarely do they cause repression. Okay, so we're talking about acetylation of histones, and you know that has to do with gene activation. Okay, so the acetyl groups, when you add multiple acetyl groups to a particular structure, in this case your histones, it always tends to destabilize your chromatin structure, which then allows for transcription apparatus to access the promoter and the gene. acetylation are done by enzymes called acetyltransferase. Very easy, okay? Transferring the acetyl group to the histone. And of course, deacetylases then are the enzyme that can reverse the effect and remove acetyl groups to suppress gene expression by restoring that chromatin structure. So here, for example, is a diagram where we have acetylation of the histone tails, okay, which destabilizes this whole chromatin structure of your nucleosome. So now instead of tightly wound around each other like that, now we have loosely wound DNA around histone molecules, and you have exposure of this particular gene, and now this becomes accessible to your RNA polymerase, transcription factor, that whole apparatus that needs to bind in order to activate gene transcription, okay? So, but if you were to remove these acetyl groups by your deacetylases, then it actually wraps the histones back up around the particular DNA sequence or gene sequence, restore that structure, and in a sense, we can repress the transcription of that particular gene when you have deacetylases happening, okay, or functioning on the gene. Now, a third way to change chromatin structure is by DNA methylation. Notice that this is different from histone methylation. Don't confuse the two. DNA methylation is different from histone methylation. Histone methylation is only adding methyl groups to the histone proteins, like these green dots here on the histone tail. DNA methylation is when we're adding methyl groups directly to the nucleotides themselves. We're talking about your adenine, cytosine, etc., thymine, guanine. Oftentimes with DNA methylation, it is the nucleotide base cytosine, okay? Usually it is cytosine that gets methylated and becomes a 5-methylcytosine. Usually more than one cytosine can be methylated for it to take effect. So that's why those DNA sections that are heavily methylated usually lead to gene repression, while DNA that are unmethylated will have gene activation, okay? So this picture here is kind of representing, giving you a glimpse of both histone methylation, which is the actual green dots, the methyl groups adding to the histone tail, or DNA methylation. As you can see, the green methyl group on the DNA itself, okay? And the more of these green dots you have around the DNA sequence itself, you will have gene repression, whereas when you demethylate these green dots from your DNA sequence, then will have more gene activation, okay? So again, now, the other thing that differs between histone methylation and DNA methylation is with histone methylation, it can go either way, activation or repression, depending on the case. But for DNA methylation, most of the time, it is actually gene repression when we're talking about heavily methylated gene, okay? The more methyl groups you have on those cytosine molecules, the more it is that gene is going to be deactivated or repressed from expression. Whereas when you have no methylation on your DNA molecule or gene, then that will become gene activation. Okay? All right, so let's take a break here and we will come back in part 2 of the lecture.